Everyone, I'm AnnieJo Yeager, and I chose to do my presentation on plant and animal mitosis and meiosis. So today we will be looking at the differences between mitosis and meiosis in animal and plant cells. So some of the questions will be what differences, if any, exist between plant and animal mitosis? What is cytokinesis? Are there differences in the way the cell walls form? What is meiosis? How does the process of meiosis in sperm compare to meiosis in pollen production? And lastly, how does it compare between egg and seed production? So overall, plant and animal cells are relatively similar in the process of mitosis, but a few key differences set them apart completely. So first, we'll be looking at the differences in mitosis. So first and foremost, it is important to consider the structural differences of plant and animal cells. The plant cell shape in general differs from the animal cell shape. So if you look at the diagrams below, the plant cell is the green one with a more hexagonal shape, and the animal cell is the cylinder one with a more circular shape. So notice the plant cell is missing an organelle that the animal cell has, the arrows pointing to it, the centrosomes. And lastly, the plant cell has a cell wall, whereas the animal cell does not. The animal cell only has the plasma membrane. So difference number one is aster formation. The aster formation comes from that missing organelle that plants lack, the centrosomes. In the plant cell, it is the reason for one of the differences in mitosis. The centrosome is made up of centrioles that is only present in animal cells. These centrioles cause aster formation in prophase and mitosis in animal cells. Aster formation is the star-like formation of the centrosome and the microtubules attached to them. So as you see below, the arrows pointing to the asters, those microtubules are the little lines that come out from the centrosomes. So a close-up look with plant and animal cells in prophase. The animal cell, you can see the aster formation around the centrosomes. In the animal cell, you can see that there are microtubule spindles, but there's no centrioles that makes a star-like formation. So cytokinesis is next. Cytokinesis makes up the rest of the differences between plant and animal cells between mitosis. Cytokinesis is the division of the cytoplasm in the cell wall, which results into two daughter cells. So if you look at the diagram below, you see the initial cell, and then it results into two identical daughter cells. The daughter cells are not only identical to each other, but identical to the initial cell. So difference number two is when cytokinesis actually begins. So in animal cells, cytokinesis begins at the end of anaphase, whereas in plant cells, it begins at the end of telophase. Cytokinesis happens later in plant cells because of its rigid cell wall. So remember, animal cells lack that cell wall, so the structure is not held up by a cell wall. So that rigid cell wall also causes the forming of the cell wall to be different. So difference number three is the formation of the cell wall in cytokinesis. So as mentioned before, the cytokinesis of an animal cell begins at the end of anaphase. It's noticeable if you look down at the diagrams below, where the arrows are pointing, the little indentation in the side, that's a contractile ring that pinches the cell into two equal daughter cells. That contractile ring is called a cleavage furrow, and the lack of a cell wall makes it where the animal cell can be squished together and pinched in between. But since the plant cells have a cell wall, the cytokinesis is a little bit different. In plant cells, the rigid cell wall prevents the cell from being pinched into two, so instead a cell plate is formed by phragmoplasts. Phragmoplasts are microtubule structures that act a lot like the centrioles of animal cells that plant cells lack. These phragmoplasts carry vesicles of material from the cell wall to create a new cell plate at the equatorial plate, or the middle of the cell. So if you look at the diagram, you see the vesicles that come from the cell wall that's surrounding it, so the big green outlining, and then it splits the cell into two by putting a cell plate in between them. So all about meiosis. Meiosis is very similar to mitosis, except in addition to separating sister chromatids, the homologous chromosomes are also separated. It is used for sexual production, and meiosis results in 4 gametes, which are sex cells, so sperm cells or egg cells, instead of 2 identical daughter cells. And those 4 cells are all different. Meiosis is a two-step process that consists of meiosis 1 and 2. So meiosis 1 is what we'll look at first. Meiosis 1 consists of prophase 1, metaphase 1, anaphase 1, and telophase 1. And as mentioned before, it is very similar to mitosis. So prophase 1, the nuclear envelope fragments, chromosomes condense, and crossing over takes place, and there is formation of tetrads. In metaphase 1, the duplicated homologous chromosomes line up at the equatorial plate, but they're facing each other, whereas in mitosis, they don't face each other. In anaphase 1, the homologous chromosomes separate, but the sister chromatids are still united at the centromere. In telophase 1, the duplicated homologous chromosomes reach the poles of the cell, but the sister chromatids are still connected at the centromere. And at this point in mitosis, the sister chromatids would be pulled apart. So here's a visual of meiosis 1. You can see the tetrads where crossing over takes place, and you can see that aster formation in the animal cells where the centrosomes are. And then in anaphase, you can obviously see how they face each other. So in meiosis 2, it consists of prophase 2, metaphase 2, anaphase 2, and telophase 2. So again, the chromosomes condense, the chromosomes reach the spindle poles and the nuclear envelope fragments in prophase 2. In metaphase 2, the chromosomes again align at the equatorial plate. Anaphase 2, this is when the sister chromatids are pulled away from each other and toward the spindle poles. In telophase 2, the chromosomes arrive at the spindle poles and they decondense and a new nuclear envelope forms. So meiosis 2 is pretty much exactly like mitosis. So here's a visual of meiosis 2, and this is where the main difference between meiosis 2 and mitosis takes place. It happens in 2 different cells, so the result is 4 haploid cells. So meiosis in animals directly produces gametes, which are the eggs and sperms in animals, but in plants it forms spores. The haploid male and female spores develop into tiny organisms that then produce the gametes. So in plants, sex cell production is aided by a process called alteration of generations. So in males, the anthers of the plants produce male spores called microspores by meiosis. The microspores germinate into pollen grains. So the sporophyte phase produces the spores by meiosis, and the gamophyte phase produces gametes in mitosis. And then in males and animals, the sperm production, the germ cells are called primary spermatocytes that undergo meiosis and produce 4 haploid cells called spermatids, and all 4 of these cells develop into sperm cells. So sperm production is followed directly by meiosis. In females, again, it's a process by meiosis and only meiosis. The germ cells are called primary oocytes that undergo meiosis and produce 4 haploid cells called ovum, but only one of these 4 ovum produce the egg. All the other cells degenerate. So seed production is the female version of more alterations and generations. So the pistil contains an ovary at its base of a flower in which the female spores called megaspores are produced by meiosis. These megaspores undergo germination and produce a gametophyte, which is what produces the seeds. So meiosis aids the process of seed production but does not produce seeds. It doesn't produce seeds directly like meiosis would with eggs and sperms. And that concludes the presentation and all the differences and some similarities between animal cells and plant cells. Thank you.